So, this is session number 2. Session number 1 covered a good chunk of material, mostly the first quarter. Howdy, howdy. Go ahead and sign in for me. And what we're going to do is we're just going to pick up and try to do basically everything in quarter 2. Once again, you're free to come and go as you need or please, and let's do this thing. So, picking up where I left off last time, I'm going to go ahead and do us a favor and move this out of the way so you can actually see. So we're going to go ahead and talk about the structure of the cell membrane. I just want to throw this phrase your way. Hopefully this word sounds familiar, but it's phospho. Oh, by the way, if you haven't been able to tell, I'm awful at spelling, and my handwriting is awful, so deal with it. So phospholipid bilayer. The phospholipid bilayer is basically 2 layers of phospholipids. Phospholipids are these molecules. They roughly look like jellyfish, and they look kind of like this. The phospholipid, we said, I know we covered this last week, but just a review. A review and a review. There's a head and there are tails. The head is comprised of the phospholipid, but we call this the head. And the tails have these, in this case, 2 tails. They have this special feature. Basically, the head is what we call polar, and the tails are what we call non-polar. This is really important because polar substances, like water, will mix together with the heads very nicely, but the non-polar tails, they hate water so much that they don't intermingle at all, which forms this structure. I'm not sure if you guys can really tell, but if you look here, there are 2 layers of these jellyfish phospholipid bilayers. So the heads are on this side and that side. So this side loves water, but this middle portion absolutely despises it, which means water has a challenge moving through there. Also, we use this other phrase. Does anybody remember that special model that we use to describe the cell membrane? It's got a funny... Blah.
I remember when we did that.
You might remember. Yeah. It's called the fluid mosaic model. Yeah. And the fluid mosaic model basically is titled that. Gentlemen, hello. Please sign in for me. Please sign in for me. Please sign in for me. Thank you. There are donut holes and orange juice behind you, so feel free. Feel free. So the fluid mosaic model, fluid mostly just means that it can move, so it's mobile. And this is pretty important because this means that the items that comprise the structure can actually move. They can respond to their environment. If it was stiff or rigid and there was force or activity pushing on it, it would break, it would tear, it wouldn't work properly. So this is really important. We want it to be fluid. Even there are conditions that if our cell membranes become hardened, they could break and tear, and that wouldn't be good. The other thing is that it says mosaic. This word's very nice because when you look at the structure here, it's actually got a lot of variation. Back in the past, people used to think it was just 2 layers of membranes, but that's not true at all. So now when we look there, you see these blue blobs everywhere. These blue blobs are all different kinds of proteins, and you can kind of see that written in there. Protein shows up everywhere. And remember, proteins are like one of those main structures. They're like a workhorse of your body. They do all kinds of things, and they have a lot of different purposes in the cell membrane. So when we look here, you can see some of them have openings that are designed, so things can move through them. You can see these are called channel proteins because they're like a tube, they're like a pipe that allow things to pass through whenever they want to. You can also see there are some other kind in there. These are called integral proteins or peripheral proteins. Peripheral proteins mostly lock in this thing called the cytoskeleton. They kind of hold it in place on the inside of the cell, and that once again is a framework for a lot of the organelles to use. The integral proteins, howdy, sign in for me please. The integral proteins, those are going to be found basically to basically strengthen the cell membrane. You'll also be able to notice there's some other goofy things like cholesterol. Cholesterol isn't always bad. There are different types of cholesterol. When it's in your cell membrane, it's supposed to strengthen your cell membrane. And then we have these things like glycoproteins and glycolipids. This means basically a carbohydrate attached to a lipid or a protein attached to a carbohydrate. These kind of act as like receptors. And recently in class, we've been talking about immune systems. And we said immunity is like your body's ability to fight off disease, right? And they got to identify the bad cells. Well, the things that identify those cells are going to be these little stick things on the outside, and that's what they're comprised of. So that's what those basically are. So the cell membrane, keep in mind, it is dynamic. It's changing all the time. It's moving. It's fluid, and there's a lot of parts and pieces in there. There have even been experiments that basically froze this and broke it in half, and they were able to see what side the proteins stick to, and that was actually really important to our discovery of how dynamic it really is, and that's pretty important. Does anybody have any questions on the cell membrane? Anything like that? For all those people just walking in, there are donuts and orange juice over there for you. Remember, you're free to leave anytime you want. If you want to chill out, if you signed in, cool. If you don't sign in, that's also still cool. It doesn't really matter. It just helps pay me one day, maybe, possibly. We'll see. All right. So that was kind of the end piece of like all the different cell organelles that we have. The cell membrane, super duper important. So let's go ahead and talk about the individual cells. Now there's two major categories of cells when it comes to eukaryotes, and that's what I'm going to go ahead and write off to the side here. Eukaryote. And just a callback from last week or just previous knowledge, eukaryotic cells and prokaryotic cells are the two different types of cells, right? Prokaryotes are dumb. We made fun of them a lot. You can like get in your stuff. It's all good, man. Like the mic probably won't pick you up. It's okay. And if it does, then I'll make fun of you later. So prokaryotes, they don't have a nucleus. That's why they're so dumb, right? Their DNA just kind of floats around and does whatever. But eukaryotic cells, those do have a nucleus. And there are two major categories of eukaryotic cells, plant and animal. So animal cells, you can see here, and I'm just going to give you a little walkthrough of kind of those organelles that are found there. These are all membrane-bound organelles, which means they are like their own self-contained unit. And since they share the same stuff that the cell membrane's made of, that allows them to merge and kind of like transfer stuff between them. So, of course, you'll see here it's written as the plasma membrane instead of the cell membrane. And that's probably the more proper term is plasma membrane. But whatever. It's the cell membrane. It's the skin of the cell. And you'll be able to see there are some cool things out there like the Golgi vesicles, which are related to the Golgi apparatus. Remember, that's like FedEx. Ribosomes, this is where proteins are made. This is a really, 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 really important organelle, and it's found in all types of cells, even prokaryotes. We've got the rough ER, the smooth ER. Rough ER is where the proteins are made. Smooth ER is where lipids are basically made. They've got actin filaments here, and those are just different kinds of protein networks that kind of go throughout there and help move things around inside of the cell. We've got peroxisomes, and lysosomes, which basically help break down things inside of the cell, kind of like the recycling center. Microtubules, another structure within the cell that gives it support. There's free-floating ribosomes, which still make proteins. Mitochondrion, so the mitochondria make energy for the cell, and of course you're going to be able to see those are very much there, and then you'll be able to see the intermediate filaments, another type of the cytoskeleton. Secretory vesicles, these just transport things, vehicle vesicle. Centrosome, this is going to help like move the cell's parts a little bit later on, and then cytoplasm is the jelly-like substance inside of the cell. Some of these have a flagellum, which means they have like a little finger or a little tail, and it helps like move around and transport these things. And of course, inside of just the nucleus, you've got basically chromatin, which is loose DNA, the skin of the nucleus, which is the nuclear envelope. The nucleus, of course, is just the whole thing, but the nucleolus is the deep kind of the core of the nucleus, and that's where most of the DNA or the chromatin is contained. Now I point all these things out to you because the main things you'd notice about animal cells is that they have only, in terms of energy, mitochondria. Okay, that's key. They do not have chloroplasts, which I know you've heard of before, but you don't see them here because animals are not green by nature. To the point where they're absorbing sunlight and to help them power, energy processes. So they can be green. I'm not saying like snakes aren't really green, guys. I'm just saying they don't absorb sunlight for photosynthesis. Also, when it comes to animal cells, I will point out they have very small vacuoles. And vacuoles, if we look up here, you know, it's not really listed because they're so small, they're almost inconsequential, and their basic job is basically to hold waste, hold junk, so that the for the lysosomes and the peroxisomes to show up, release some enzymes, and break those things down. I should also go ahead and say there's another big feature on the outside of this thing that's not there. What is that?
Isn't it the cell wall?
It sure is. It's the cell wall. The cell wall is not present, and that's never present in animal cells because animals got to move around. That's what they're known for doing. So if there was a cell wall in these cells, it would be so tough that they couldn't be mobile. The cell membrane would still function and do its thing, but it wouldn't allow for the free movement. But when we look at plants and stuff like that, they are dependent on the structure of the cell wall to the point where, you know, it prevents them from moving around or at least growing very quickly. So those are the main differences between animal cells, things you should distinguish for animal cells. Any questions there?
Is the plasma of the plasma membrane, is that the same kind of plasma that's found in like blood cells?
Oh, no, it's not. It's interesting that we phrase it that way, right? But in blood, plasma is mostly just like a water content. Of course, there's proteins floating around. Plasma membrane, you know, honestly, I don't know the reasoning behind why they choose some of those words. It's interesting, though. So, all right. So we've got plant cells. Once again, I'm just going to point this out to you. These are also eukaryotic cells. Once again, guys. Hi, bye.
Bye, Mr. Olson.
Eukaryotic cells, once again, have a nucleus. So when we look here, we can definitely see there's a nucleus in place, right? I should change colors so it makes it a little easier to see. I don't know if it'll help, but whatever. Let's try. Is that better? She was really proud of herself, wasn't she? She like rolled in like, Anybody listening to this video is like, I have no idea what's happening, but that's fine. So the nucleus is still in place, and it still has all the same parts and pieces that you're familiar with. They still have the same organelles, though. Ribosomes, still the same. Smooth ER, rough ER is still there. Where'd you go? There you are. The Golgi apparatus, the Golgi body, whatever, the Golgi complex. It's called all kinds of things, but it doesn't really matter. Golgi vesicles, cytoplasm, peroxisomes, mitochondria. One thing kids usually forget about is that mitochondria are still in plant cells. I know kids think chloroplast plant, mitochondria animal, but the truth is plants got to have both. This is really a plant world. We don't need to be here. So plants can literally survive by themselves because they can break down the sugar they make from photosynthesis. They can break down that sugar and survive because they have mitochondria to be able to do that. They still have the plasma membrane. They still have like the cytoskeleton. They still have some vesicles and all that stuff. But there's a few distinctions. First off, we see chloroplast here. So chloroplasts. Am I spelling it right? I think so. Yay! So chloroplasts are present. Okay, that's a big distinction. Once again, they're not in animal cells, but they are here. And they're filled with this stuff called chlorophyll, which is like this pigment that actually absorbs sunlight and helps them convert that into another process. You can also see there are vacuoles listed here. The reason for this is because they have a large central vacuole. Now, a large central vacuole is really important to plants. The reason for this is because this is where they store a ton of water. And plants are taking in water all the time because they're going through photosynthesis, right? And one of those main ingredients of photosynthesis is water. So they got to have a lot on site. The other thing is, since they basically don't have a bone structure, they don't have, you know, a skeleton, what they're basically doing is they're filling up this balloon inside with water, and water doesn't compress. So they can get really, really, really big, really heavy, really massive in terms of plants, and all that weight will just try to compress those balloons, and if it does it equally, it actually helps retain the shape of that cell. Turgidity is the word we use to describe that. And then the other thing that you'll notice is, of course, the cell wall. These guys have a cell wall. This is also true of fungi. Fungi have a cell wall as well, but their cell components are a little bit different here. Instead of just making a cell wall made of, in this case, for plants, I'm just going to go ahead and write down the word cellulose. And if it ends in OSE, it tells you it's a sugar. So this is a type of sugar, but in fungi, theirs is made of chitin. It's a protein structure, and it kind of gives mushrooms that rubbery texture on the outside. Kind of feels like gross rubbery skin. You know what I'm talking about, when you bite it and the mushroom squeaks against your tooth. Yeah, that's chitin. That's causing that. So cellulose, you mostly know as like fiber in your diet. Our bodies don't process that sugar very well, so when you chew it up, you poop it out, and that's actually what helps you pass so much stuff through your poops. I'm saying poops. Poop. All right, so any questions on plant cells? Feeling okay? Yeah? Okay. So if you know about those plant cells, then we get to plant structures. So let's go ahead and look a little bit closer. Let's look at like a mass of those cells working together. This is going to be a leaf, okay? So this is going to be a leaf dissection. And what we're seeing here is going to be a few layers of cells. Nothing too crazy, but the main things I want to point out to you is that we know cells have to use some cell processes, right? We talked about cell respiration and photosynthesis, and we'll go more in depth here in a minute. But let's just get like the components down first. Well, they have to get oxygen and CO2 in and out of these cells, right? So how do they do that? So in a leaf, which once again is really wide, like look how wide that is. It's designed to absorb sunlight like a solar panel, right? And then all of these components inside of there, especially these little green spots in there, those chloroplasts, they're doing the work of actually absorbing sunlight. So you can kind of see there's our cell components, right? You'll notice that there are a few layers here. First off, we have the cuticle, which is kind of a waxy layer. It kind of lets water run off of it, so it just doesn't penetrate the cells over too much. Then we have the upper epidermis, which is basically their skin. So there's a thin layer of cells, and it's usually just 1 or 2 cells thick. And then we see this middle region, which we call the mesophyll. Meso means middle, so we're saying about the middle cells. And then there's the palisade mesophyll and the spongy mesophyll. Palisade means like it's really packed in there, but spongy means literally like a sponge, there's open cavities. And those are really important for moving around those gases we talked about. So all plants will go through this process called transpiration, which means they have to move water vapor and they have to move oxygen and CO2 out of the plant itself. Now water obviously will move into plants by the roots and all that stuff, which I really hope you know what stems, roots, and leaves and flowers and are. Like, I really hope you know that. If not, you're scratching your head a little too much back there. So you'll notice that there's gases that are going to open through these openings on the bottom. We call these stoma. For plural, we call it stomata. But anyway, it's surrounded by these guard cells. So basically a guard cell, oh boy, I'm a very, very good artist. I take it all back. It looks kind of like a donut or 2 macaronis. Wow, look at me. I'm such a good artist. But these are guard cells, okay? And then there's the opening here in the middle. That's the stoma. The stoma is literally just an opening, and these things open and close based on how much water is filling up those cells, and that's basically the thing that allows us to release water vapor and release oxygen gas and bring in CO2 gas. Because CO2 is a gas. You're not going to find that like pulling that out of the ground, right? That's not a thing. And then, of course, we have the very lower limits, the lower epidermis and the cuticle. Just try to keep in mind that spongy area allows for those cavities to allow gases to move through. And that's basically how that integrates into the leaves. And that's how photosynthesis is facilitated. Ooh, big word. How it's helped along. So do you guys have any questions on the leaf structure? No? Remember, leaves, stem, roots. You know what the roots do, right? I hope so. But they absorb things from the soil, namely water, right? A lot of nutrients in the soil like nitrogen, phosphorus. And then, of course, they help root down the tree. They help anchor it to the ground or whatever plant it is. Because if there's a little tiny breeze and there's no roots, then it falls over and it dies. So it's got to stay in the ground, right? The stem, of course, is going to support everything. It's going to deliver things from the roots up to the leaves, and the leaves are basically where photosynthesis happens. That's why leaves are typically green, because their job is to just absorb a lot of sunlight. And we even talked about, like when we talked about ecology and we talked about like rainforests, they have really thick canopies of leaves, and sometimes their leaves are massive, and that's literally just to get even the most minute amounts of sunlight into the plant and make it as efficient as possible. Doing all right? Yeah? Any questions? Are you sure? Okay. I think we're good. Let's go ahead and talk about photosynthesis. I'm going to hang here for a minute. I'm sorry, but photosynthesis is pretty important. So let's go ahead and look on the left side here. And we're going to see basically this little round blob, this kind of bean shape. This is going to represent a chloroplast, okay? And in our chloroplast, we have a few structures. So let me go ahead and write those words down. We, of course, have the chloroplast. We have this thing called, I hope you remember this, but these are called grana. And these are made up of individual thylakoids. Do these words sound familiar to you? So a thylakoid is going to be like a green little pancake that sits inside of the chloroplast, and the stack of pancakes are called the grana. And then there's this open space that's inside of the chloroplast. That's called the stroma. Not the stoma, even though I know we just mentioned a word that's very similar. It's the stroma. It's the open space in between the thylakoids. This is the area, this is the factory that allows photosynthesis to take place. So what I want to do is I want to kind of divide this without cutting through any of the words here. I kind of want to divide this in half, okay? There's a front half and a second half, okay? So we're going to look at this in two capacities. I also just want to go ahead and mention, if we got some of this terminology out of the way, I just want to throw this one out there too. ATP. Do you guys remember ATP? Adenosine triphosphate. It's basically a molecule, and I'm just going to very simply draw this. It's a molecule that has 3 phosphates attached to it, right? The whole purpose of our ability to move around is like that we have energy, right? And the most common form of energy we use for cellular processes is going to be ATP. So it's got 3 phosphates attached to it. But if we ever want to actually get energy from it, there's a cycle that it can take place in. I'm going to draw this again. But we turn this ATP into ADP. So ATP to ADP. All right? Now, basically, all we're doing here is breaking off a phosphate molecule. When we do that, which is basically called phosphorylation, when we break that one molecule off, not even molecule, when we break that atom off, we basically end up releasing a bunch of energy. So that can be used by, whatever cellular process we're trying to go through. If we ever want to rebuild ATP, well, we already have ADP. We just add a new phosphate. That's the basic concept. So for photosynthesis, cells are trying to make some ATP, but they don't make a ton. Really doesn't make a lot of ATP here, because their whole goal is to turn solar energy into chemical energy. So they're trying to turn sunlight into sugar. And then later they'll break down that sugar and get a lot of energy from it. Because the other process that is really involved is cell respiration. And that stuff breaks down sugar and makes tons of energy like crazy. So let's go ahead and look at this thing on the left. On the left-hand side, we call these the light reactions, or the light-dependent reactions. They depend on sunlight being there, or just any light in general being there. So, of course, the key ingredient is the sun, is the light, and that's going to be received in what we call basically these 2 photosystems. Do you guys remember talking about those? The different photosystems? I don't know if your other teacher ever went into it. Yeah, it's kind of goofy. So I'm going to now direct your attention. We're not trying to detract from anything, but I need to direct your attention now to picture number 2 over here on the right. You'll see where it says PS2 and PS1. As simply stated as I can make this, because it could get really messy and complex. photosystem 2 happens first, right? And this is where sunlight comes in, and then this chlorophyll, which is this molecule that you see right here, that's chlorophyll, or a type of chlorophyll. It gets all activated, it gets all excited, oh, it gets all happy, and when that happens, electrons want to start moving. The other cool thing that happens here is water, stuff that's already in these plant cells, it starts getting broken up. When it breaks up, it releases O2, okay? And that's just a waste product of photosynthesis. And it just gets broken off really early on. But then you'll notice it has these hydrogen ions. They're positively charged hydrogen ions, which in truth just means it's a proton. But whatever, that gets like stuck inside of the thylakoid, and this helps like create this electrical gradient on both sides of the membranes. Anyway, once this happens, you'll see that some electrons are going to want to transport, kind of in a long, snaky way. They're going to want to start transferring through here, right? And then we hit photosystem 1. Once again, photosystem. So photo means light, so more light's added here, and another molecule's all excited. And basically, this pushes some more electrons through there, and then we build up all this stuff. Now, once again, I'm cutting corners here, but see that molecule ADP? We want to turn that into ATP. So in this case, we're making just a little bit of ATP. It's not a lot, but you'll notice the buildup of all these hydrogens that are forming over here. When they pass through this molecule called ATP synthase, they're making ATP. So it does make a little bit of energy. But the other thing is, we've just done the first half of photosynthesis, which means now we can carry all this leftover stuff and make something happen. So we've made a little ATP, which we see here, and we've made something called NADPH, which I don't know how on the EOC they'll be about that molecule, but basically consider it like another version of ATP. It's a high energy molecule, and it can basically be spent. So to recap that first half, water and sunlight are brought in. The byproducts are oxygen gas, which literally gets released out, and that's what we breathe. Thanks, plants. And then, of course, there's a little bit of ATP and NADPH that are made. Now we go to this thing called the Calvin cycle, the second half of photosynthesis. When I teach this to you, sometimes I refer to this as the light-independent reactions. Do you guys remember that phrasing? So sunlight's not directly needed for this to work, okay? But of course, the first half of the process needs to occur. So at some point, sun does need to fall on these cells. Anyway, so this energy's brought in here, and through this very messy, complicated cycle, we basically turn some really simple molecules. We bring in CO2, and we basically build up these complex 6-carbon sugars. We break them down, and we steal away some molecules called pyruvates. I'm just going to point out to you, this says glucose is the export. It's not really glucose, but that doesn't matter. Like, the basic thing is this, and I'd say all of this to get to this main concept. It's crazy, I know. So what do plants need? They need sunlight. They need water. They need carbon dioxide. I'm looking at it now like this. They need this, and this makes oxygen gas and sugar, which is C6H12O6. Okay? That's the formula for photosynthesis. If you know the formula for photosynthesis, you should know basically everything that would really pertain to the EOC. I've seen like plenty of sample test questions where people have looked at questions about photosynthesis, and if they always go back to, oh, I know the formula for photosynthesis, then they can kind of figure things out. They can even stumble across the answer sometimes. But you should be clear, that's the formula for it. Whew. I know, right? How you doing? Do you guys have any questions there? Or praise? I'll take praise. Tips? No? Judging me with your silent eyes. I see you.
Are eyes supposed to speak?
Mine talk all the time. My eyes speak
Definitely see a doctor.
constantly. It makes sleep very difficult. All right. So let's go ahead and talk about the other process. we've got cell respiration. So I'm just going to go and distinguish right here. I wrote the word aerobic. Aerobic cell respiration, there's another form out there called anaerobic cell respiration. Right now we're talking about the big money maker. We're talking about aerobic cell respiration. Once again, this would happen in a molecule called mitochondria, or the mitochondrion, but mitochondria for plural. So what we're looking at here, another kind of kidney-shaped, bean-shaped organelle, but you're going to see there's like little folds on the inside. We're basically just trying to make sure you understand it's happening in this particular organelle. So let's go ahead and look at this, and this is broken down into basically 3 parts. So we can look at it kind of like this. Okay. The first thing that's got to occur is glycolysis. I'm just going to go ahead and like make a shortcut here, and I'll explain that more in a second, because we'll talk about anaerobic stuff. But this is happening outside of the mitochondria. It's happening here, so it's not happening inside of it. But basically what happens is we're turning sugar into these molecules called pyruvates. These pyruvates are then fed into what we call the Krebs cycle. Now you'll notice some energy is made here, NADH. That'll show up later on. But anyway, our goal is to shuttle electrons. Now, I don't need to look at the membrane for this, but it's basically the same thing as the photosystems, photosystem 2 and photosystem 1. Just in this case, there's no sunlight involved. So it's basically just moving across an electron across a gradient. And when we do this, we can have a lot of ATP made because it passes through that molecule ATP synthase. So anyway, first thing we do is we go into the Krebs cycle, another complex process. This is also sometimes called the citric acid cycle. Okay? And this basically takes in these pyruvates, a little extra energy from what's out there, and this fun molecule called acetyl coenzyme A. And this basically works through a complex process of which you don't have to know any of the molecules of. And it works through there, and it spits out just a little bit of energy, but it also spits out more of another kind of energy. All of this basically gets fed into the electron transport chain, which is just like those photosystems that we talked about. It basically has that complex marching along process. But notice the words that they use here. Oxidative phosphorylation. Phosphorylation is attaching phosphates to this ADP. So we're turning ADP into a lot of ATP. So if we look at the end results, we'll see this produces about 34 ATP per turn. That's a ton. Because in the last thing for photosynthesis, we saw like 2 getting made, and then they get used up just to make sugars. And then in this process, the breaking down of those sugars, we make at least 34 at the end of this. This does make a little bit of ATP earlier, and of course glycolysis makes a little bit too. And we'll be able to talk about this, but total, there's about 38 ATP that are made in this process. That's a lot of energy. It's pretty darn efficient. It's not perfect, but this is what we've evolved to use as our main form of energy processing. So I've kind of said all this once again to now bring up the formula for cell respiration. This means animal cells or even plant cells, they need to take in sugar, so C6H12O6 plus oxygen. They have to take in oxygen, and then they release CO2, a little bit of water, and of course energy, usually in the form of ATP. This is the simple formula for cell respiration. Now let me just point out to you, this is the almost exact same thing as photosynthesis, but it's reversed. So if you take all this stuff and you move it over here, and you take all this stuff and you move it over here, and you still have your arrow, all you got to do is change that to sunlight, and then it still says CO2 and still says H2O, and this still makes sugar and oxygen gas. You have photosynthesis. So in a lot of cases, there are test questions that basically say things like, hey, where did the carbon for the sugar come from? Well, it came from the CO2 that was found outside in the environment. they take in CO2 and they break that down and they turn it into carbon in sugar form. And then that's eventually broken down through cell respiration to make energy. So they may ask you questions like that. So having a good understanding of the relationship between these two different processes and how they're really closely related is probably the key to answering almost any question they could ask you in the EOC about this stuff. Doing okay there? Yeah.
Okay. What is going, you said it like 2 seconds ago, what is going into the Krebs cycle? Like, just to clarify.
So I should mention, yeah, I guess I should just mention this. That would be oxygen. Oxygen is brought in, and I believe we spit out some H2O at this point. And then we do bring in a lot of other goofy things at the beginning of this too. But yeah, the sugar is basically here, glucose, that's sugar. That gets broken down, and then we're able to process that better. We bring in some oxygen here and there. We spit out H2O, and then of course we spit out a ton of ATP or energy here. A little bit better? Mm-hmm. Sorry, I didn't write that earlier. Any other questions? No? All right. So I mentioned glycolysis, right? And I just want to be clear, there's something special about glycolysis. It is an anaerobic process, which basically means it will function in a no oxygen, no O2 environment. That's the key feature. As a matter of fact, if I'm phrasing this correctly, it won't really occur if there is oxygen nearby, but it's very necessary for those oxygen necessary processes. So when we look at this, we said the molecule is C6H12O6, right? If this molecule were to get fed into the Krebs cycle, for instance, this is way too big. It's a big, ugly, complex molecule, and it's got 6 carbons. Think of it like a hexagon. That's really complex. So what the body wants to do is basically break this in half. So we turn this into 2 molecules called pyruvates. And these are kind of lower, structures, smaller structures. They only have 3 carbon each, so 3C instead of 6 Cs. And also, I should mention, this process does some interesting thing is while this happens, it produces 2 ATP. Now you're like, you're devoting a whole slide just to this goofy little process. You already had words written up there earlier, man. What's going on? Well, 2 ATP is just barely enough positive gain in energy to make something survive. A single cell can survive pretty easily on this process, which leads me to anaerobic respiration in terms of the 2 different processes. So there's 2 different forms we care about. One is called alcohol fermentation, and the other one is called lactic acid fermentation. I'm gonna get lazy. So if you're one of my kids, you probably already heard my story about lactic acid fermentation, right? When I was in 9th grade, I worked out for the very first time, and my muscles got all painful, and I was like, ew, I can't pick up the remote, right? That's what I did as a 9th grader. I was so smart. But it's no joke, because lactic acid is a surprisingly effective molecule, and it sends a few signals to us. But let's go ahead and walk through this process. First off, lactic acid fermentation, glycolysis happens first. In both of these processes, glycolysis is going to occur first. But in glycolysis, we get pyruvates. Okay? Pyruvates basically go through a small change, and in this case, that 3-carbon molecule, it's turned into another 3-carbon molecule that we call lactate, which is lactic acid. And this is the stuff that spreads around in your muscles and causes that pain after you exercise them, or if you've ever been running for a long period of time, you get that stitch on the side, and you're like, ouch. Well, that's a little buildup of lactic acid. If you've ever seen people like process cheese or something, and it's like fermented for a while, they're probably using some lactic acid to help, like, make that a little bit more bitter, But anyway, this is like really important in the human body, or just bodies in general. That's where this is found. But the most profitable biological process on Earth is probably alcohol fermentation. I'll give you one guess on what it actually makes. Good try. It is alcohol, though. It is alcohol. So in this case, we have glycolysis. And then when we do this, we still make pyruvates. So once again, that's a 3-carbon molecule. Now in this case, something different happens. This breaks off into 2 different things. The first is a 2-carbon molecule that we call an alcohol. And if you've ever heard of alcohol, there's a lot of different forms of it. There's maltose, there's lactose, there's, oh my gosh, I don't know, man. There's like so many, and they're all like used for different purposes. But they're just 2-carbon molecules, and they're called alcohol. But where did that 3rd carbon go? Well, it breaks off in a molecule called CO2, which is why whenever you see images or a video of like people like popping a bottle of champagne, which is an alcoholic beverage, there's a whole lot of foam created because all those CO2 bubbles are now becoming, they can escape, they can run away, which causes that foam over. This is like a big deal. This is really important because it shows us in this chemical process we can have alcohol as a byproduct. This happens in single-celled organisms. Typically. It can happen in us a little bit, and our body will process that. But if you ever see like fruit that is rotting, and they say it's fermented, it's because it's gone through alcohol fermentation. Sometimes to the point where some animals will eat fruit that is fermented, and they'll basically get drunk because there's so much alcohol left behind. So CO2 is released. We love using this for our purposes for humans in like breads, in like pizza dough, because you get yeast, which are single-celled organisms. You feed them sugar, you give them water, they awake, they nom 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 on that sugar, and they burp it out as CO2 and alcohol. Well, the alcohol you cook out of there. That's why there's that kind of alcoholic smell to like a bakery or something. And then, of course, the CO2 makes the bubbles in the dough, which we bake in there, and makes it all soft and plush and delicious. And I want pizza tonight, all of a sudden. How you guys doing? It's even used in making donuts, all fluffy. Huh? See what I did there? Because I gave you donut holes earlier, and that okay, you don't care. So, that was basically everything for that. Do you guys have any questions on or anything there? No? How are we doing time-wise? Not bad. So we're going to talk about some cell division. You guys ready? Yeah? Yeah? I know this kid. I know what you were doing last night.
What was I doing?
You were eating cereal at midnight.
No, I didn't eat cereal. I didn't eat cereal in like 2 weeks.
That's sad life.
I've never heard a kid lie so bad. All right.
Okay, I ate Captain Crunch last week.
Oh, interesting. I called him out on a lie, and then he owned up to it. But
No,
still,
I just remembered.
yeah,
Okay.
yeah. So, cell division. I think it's pretty obviously what we're talking about here. We're basically talking about how cells want to split, right? Cells need to make more copies of cells, and we're going to use some processes to make this happen. I'm just going to go ahead and mention this. There's some words that we care about. First one is called asexual, and the other one is sexual. I know right now, or most recently, we've been talking about sexual reproduction, which obviously means like there's some differences that can occur to make cells unique. But asexual means it doesn't need a partner. It's done solo. And this also means that we're trying to make copies of cells. So let me walk you through the most simple form of cell division that we like to call binary fission. Now, binary fission is used by prokaryotic cells. Really simple cells, and we said they're missing a key feature in their cell structures, right? We said they're missing a nucleus. Now, the nucleus is like, kind of contains the DNA, but also just makes sure that things don't get out of hand, but also just protects it from anything that might be invading the cell. It's another barrier. So with binary fission, and in prokaryotes, we basically have something that looks like this. Here's a cell, and here's the DNA. I know, I'm the best artist in the world, but that's what it looks like. I'm not kidding. There's just DNA in like a string just kind of floating around like wet spaghetti. okay? It's not contained, it's not housed, it's just there. So basically in binary fission, the cell will grow and the DNA will be copied. Let's add a new color in here. That's the exact same copy. I know I drew it exactly the same. The red and the black are exactly the same. So it's made a copy. That's a key feature of this process. And then what's going to happen is we're going to stretch this cell in different directions until basically it pops. And when it pops apart and pulls apart and breaks right there, we have one copy of the DNA over here and one copy over here. That's it. There's no special names. There's no stages. There's no phases. It's simply copy, stretch, pull. I hope it worked out. And that's prokaryotes in a nutshell. They're really, really simple in structure. They're really simple in their behaviors. And they're really simple in terms of their replication. So binary fission is key to identifying the main way that prokaryotes reproduce. How you doing? So mitosis, once again, this takes place in eukaryotic cells. And eukaryotic cells, once again, have a nucleus, so they have those organelles, and this makes it more complex. It makes it more messy for them to divide, so they have to be more precise. That's why they have these stages and phases. I'm also going to point out to you, this involves something called the cell cycle. I'm trying not to like artificially hit the button, which messes up my whole thing. So the cell cycle roughly looks like this. Here we go, circle time. It's not bad. I'll take it. So thank you. Golf claps. So when we're starting off, we're going to start off with one cell, right? We want to turn this into 2 cells eventually. That's the goal. So when we got one cell, we're going to basically break this down into some major steps. We refer to these as phases, all right? So the first phase, this is going to kind of look like a peace sign, but we're going to modify it a little bit. The first phase is called gap 1. Do you guys remember learning about gap 1? Gap 1, when you hear the word gap, you should think growth. Because that's basically what's happening here. The cell is really tiny. It's a baby cell. Oh, it's so tiny. It's so small. And it's got to grow up and it's got to mature. So in this period of time, that's what it does. It continues to grow and it matures. It'll eventually reach this area right here. Okay? Right before the end of gap 1, it's got to build up these chemicals. I'm just going to throw these words at you. CDK, or, oh my gosh, like CDKs like kinesins and cyclins. Do you guys remember those? No? Cyclin and kinases. Let's just put it this way. Maybe you remember this analogy. Do you guys like, you know, like video games, right?
Yeah.
You got to build up XP to level up, right? Right? You got to build up your experience. In some games, I get it. I understand. But let's say you got to build up experience. Well, that's like a checkpoint, right? So if you build up these chemicals inside of a cell, that gives you like approval to move on. So once you have enough of these things, you're allowed to move on to the next stage. So gap 1 is just growth. And once the cell's mature enough and it's leveled up enough, it's allowed to move on to the next phase, which we call S phase, which stands for, yeah, synthesis. Synthesis phase is basically where we do cell division. Everything we want to talk about right now is really happening during S phase. Now the cell doesn't do anything other than divide its cells. Well, not divide its cells. It's going to prepare for cell division by copying its DNA. So it's really DNA replication. Ugh. Handwriting is awful today. Okay, and then we go through gap 2. And gap 2 is more growth. The cell will continue to grow. But then we get to this region right here. And this is what we call M phase, or mitotic phase. This is when the cell goes through mitosis. This is when the cells actually split. And then the very last thing, let me go ahead and squeeze this on there. I know I can. right there at the very end of this, it goes through cytokinesis. Cytokinesis is the cutting of the cell's cytoplasm. So the nucleus divides and moves. The contents of the nucleus move, but the cell itself is still a blob that starts to get stretched out and then finally breaks apart. I should also go ahead and take the time to mention, what? Let me go back to this color. At the end of synthesis, there's another checkpoint, and just before mitosis phase, there's another checkpoint. It's checking to make sure it's got the experience, it's got the parts, everything works, so they can move on to the next and final phase and hopefully make 2 cells. There is a weird thing that can happen. Ooh, let's go dark blue. During gap 1, cells can sometimes hang back in the cycle. We call that, yes, gap 0. This is like only for some kinds of cells, like your liver cells or your brain cells. They don't divide. They chill out and stay that way, unless something were that bad happened to your liver, or for your brain, they don't actually move out of this. So gap 0 means like they're just going to stay permanently in gap 1. They'll grow, they'll be mature enough to function, but they won't eventually like make copies of their DNA or do anything special. So gap 0 explains a lot of that behavior. So, do you guys have any questions on the cell cycle? No? All sounding familiar? Okay, so knowing this, let's go ahead and pick up talking about this. First things first, we have interphase. Now, interphase is basically the name we give to everything from G1 all the way around to mitotic phase. That's interphase. So in general, this just basically means the cell's just doing what the cell's supposed to do. That's what your cells are designed to do, right? They're supposed to keep you functioning. They're supposed to do their particular roles. That's what happens during interphase, which is a huge majority of a cell's life. But anyway, there is a very interesting instance here of gap 0. But anyway, so we go through synthesis stage, we do that, we go through gap 2, cool, and then we finally follow around and we hit these stages up. So I like this image because it just kind of condenses a lot of the information in there, but I want to elaborate a little bit better on what's happening there, maybe better than what even the pictures are saying. So first things first, the first step in this process is called prophase. So during interphase, the cell's DNA is all kind of packed in the nucleus because it needs to be protected. It needs to be held in there. But during prophase, it says the chromatin condenses into chromosomes. Chromosomes are these X-looking shapes. Do you guys remember those things? So those are the chromosomes. They're not like crossed like this. They're more like this. There's 2 halves, and we'll talk about that later. Those 2 halves, they basically are going to need to split because they have 2 copies on both sides. So in prophase, the nucleus has like basically disappeared. It's phased away and just kind of goes into the ether, and those chromosomes start to move around. There's this kind of in-between stage, like I mentioned this only when I was teaching it to you, but I don't like give you super details on it, but this one does. Basically, the nuclear membrane is totally broken down, and then we see these kinetochores. Basically, these spindle fibers will start to grow from these centromeres that are going to start moving towards the poles of the cell, towards the north and south pole, and they're going to start stretching out. The DNA, the chromosomes, they're going to start moving out too. They're going to start spreading out from wherever the nucleus was. And then when we reach metaphase, you'll be able to see there's this line that forms, okay? And this line that forms, it's a straight line, and all these spindles, they start stretching across, and they start attaching to each of those individual X's on both sides, right? And when they're locking on, they're holding on to the middle portion of the chromosome called the centromere. And the centromere is like the belt on these things. And eventually, these 2 halves are going to pull in opposite directions the middle, and that's going to split it, and they're going to move to opposite sides of the cell. So we'll see them moving this way, and this way, and this way, and this way, and that's called anaphase. So metaphase is known for having the metaphase plate, which means the chromosomes are lined up in the middle, and anaphase is known for splitting them apart. After that, we're going to start dividing the cell in the middle here, And we do see this almost form, but if there's still a little connection right here, we refer to this as a cleavage furrow, by the way. There's still a connection there. We need to sever it. That severance is called cytokinesis. Okay? So that's what's actually occurring. So that's the order that it goes in, and I'm going to write that down for you in a short order. First off, I for interphase, P for prophase, M for metaphase, A for anaphase, and T for telophase. Now, if you're one of my kids, you're probably looking at this and going like, yeah, I know what this is. But if you don't remember the stages of mitosis in order, the key phrase is, I peed on a mat.
My mat.
See?
You did that to me.
I did. In your class, I peed on your mat. So I mean literally, like if I go to your bathroom, right, and I come over to your house because you like me, you want to invite me over for dinner tonight, and I get it, I understand, we're going to have pizza. I am. And I come over, and I'm like, hey, can I use your bathroom? And you're like, okay, sure. And I spend like an inordinately long amount of time in the bathroom. And then I come out of the bathroom, and I'm holding your bathroom mat, your floor mat, and it's covered in urine. And you're like, what is happening? And I say, I'm sorry, I peed on your mat. See? If you know that goofy, ugly story, you're going to remember the stages because I, P, M, A, T, C. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis. That's the order in which it occurs. So don't forget that. On the EOC, they could very easily ask you, hey, which of these stages is out of order? Oh, look at this image of the cell. What stage does it seem like it's in terms of mitosis? So knowing this stuff would be really important. And just also remember, gap 1, synthesis, gap 2. They go in that order too. You guys got any questions on mitosis? You sure? You positive? Yeah? All right. I also gave this analogy of a spider ripping in half, children.
Oh, yeah.
But, yeah, the spindle fibers are like giant spiders. 2 giant spiders on opposite sides of the forest. And they're going to stretch across and find these little kids, and these kids line up to try to get away from the spiders, and the spiders grab it on both sides and they rip them in half.
I love that.
So it's anaphase because they're getting ripped in half. I know, right?
I
Fun.
wrote that on my index cards for the midterm, and my mom was helping me study, and she's like, this one just says spider legs that rip babies, and I'm like, yeah, that's it.
Got it. Anaphase.
Got it, cool.
Understood. And your mom's like, all right, my daughter's got a problem. But nope, she was just listening to her teacher who has a problem. So let's go ahead and talk about meiosis. It sounds very similar to mitosis, I know, but it's not. Hey, just sign out for me. Thanks for coming, guys. So meiosis, it's once again going to be occurring inside of eukaryotic cells, but this one is unique. It's not the same as mitosis. As a matter of fact, in this diagram here, you will see that it says it is not a cycle. Mitosis, when it makes 2 new cells, each of those cells independently can reproduce and make their own cells. Mitosis is also asexual. This is a form of sexual reproduction. So this is how sex cells are made. And if you guys remember recently talking about eggs and sperm, hey, that's how they're made. They go through meiosis. Yeah. So first things first, they go through interphase like normal. And we'll kind of do this on the top like we did last time. Interphase still occurs. Gap 1, synthesis, gap 2. But some very interesting things happen in this stage. After interphase, and they're all lined up and they're all doing their thing, they line up and they go through prophase 1. So I'm going to go ahead and do P here, but P1. Okay, now prophase, these chromosomes that we like to call sister chromatids, but they're going to line up and just lay on top of each other, literally matching each other like twins. And they're pretty much the same thing, but there are slight differences. Now what they're going to do is they're going to do this thing called crossing over. Do you guys remember crossing over? Just in case you don't. Crossing over. I almost spelled ova. But anyway, crossing over. If we have a blue chromosome and let's say a red chromosome, they're going to literally like hug each other so close that by the time they pull apart from each other, the red one, I don't know how to do this. I just have to go back and forth, I guess. When they pull apart, there's going to be sections that are going to be different colored. And they're just exchanging that information with each other. I gave the analogy in my class, like I have my weird aunt that maybe smells like animals a little too much, and she has a lot of cats, and you know, she's always excited to see me around Thanksgiving, and then when I show up, she's like, oh, and she comes up and she hugs me, and then she's covered in cat hair, she smells like an old lady, and by the time she's done hugging me and she walks away, I got old lady makeup on my face, I got cat hair on my body, I smell like old lady. You know, my aunt's left a little bit of herself behind. She's hugged me so close, she's left some of herself on me. That's crossing over. A little bit of genetic exchange creates diversity, creates uniqueness in these chromosomes. So that's what's going to be happening here during prophase 1. So we identified crossing over. The next thing that happens is called metaphase 1. Now another very interesting thing happens here, and I know you can kind of see it in this diagram here, but the blue and the red, we have a large one and a small one. You have a variety of chromosomes. For you and I, we have 23 pairs of chromosomes, so that's 46 total. But they pair together because they're the same size, okay? And there's different genetic information on the different chromosome sets. In this case, there's only 2 chromosome sets. Whatever, who cares? But you'll see that they line up kind of like what they did in metaphase previously for mitosis. But notice how it's not a straight line here. They're paired up to each other. We call that a tetrad. And eventually these tetrads will split. So as you can see, they're separating here. And they're moving to opposite sides. But they have a full set of genetic information here. This is like super odd because they haven't lined up all the way. Interesting. So as they've split apart here, we end up with anaphase 1, and then telophase 1, and then you'll notice it goes straight to prophase. There's no back to interphase here, so it goes to prophase 2. We call this process, when they separate in this way, we call this independent assortment. This is another feature built into our cells that makes sure we have uniqueness in those cells. So crossing over isn't quite enough diversity, but independent assortment arranges things to make sure everything is unique when it gets to a parent. And also, the way this split apart makes sure that as we go through prophase 2, metaphase 2, anaphase 2, telophase 2, by the time we get here, we're splitting this again, which creates 4 cells. Now, I'm just going to point out to you 2 words. First off, we have at the beginning of this diploid cells. Diploid means they have 2 full copies of information, okay, of genetic information. By the time we're done with this, we end up with 4 haploid cells, okay? 4 haploid cells means they contain half the information that they actually should. So instead of having 2 double sets, we take the double sets and separate them to 2 full sets, and then we give them half sets each. Now this is really important because these later become sperm or eggs. And you can see here, even they're mentioning the word zygote, because a zygote is a combination of sperm and egg. And remember, mom has half your genetic information and dad has half your genetic information. Crossing over an independent assortment made sure some genes were pulled from dad, but it doesn't mean you have every trait the same as dad. So they basically get kind of randomly mixed together, crossed together, and then we form the new organism that you are. That's why we don't look like all mom or all dad. We're like a combination of both. You have mom's ears and dad's teeth. You know what I mean? And it's because of that recombination. And that's what it's actually called. It creates what we call genetic recombination. Once again, variety from something that was the same. So, how you doing? Is that an actual, like, I don't know? Or is that more like a, I just make a goofy sound for you? I don't know what that means either. Like an okay symbol? Okay, you're looking through it and I was like, does he have a telescope or something? Does he have a spyglass? All right, cool. So, let's get to genetics. All right. I know this can be pretty messy. We're going to try to keep it as simple as we can, but let's go ahead and define a few things, like what traits are, what we know about DNA. We'll clarify chromosomes and all that jazz, and then we'll get to actually doing genetics, including pedigrees, which should be fun. So let's go. Let's do this thing. You ready? Okay. I think I can do it, timing-wise. I think I can pull it off. So we mentioned this stuff called a chromosome earlier. We also mentioned this stuff called DNA earlier. Just to be clear, DNA stands for deoxyribonucleic acid. It's actually deoxyribonucleic acid, but whatever. DNA was discovered in the 1950s by these group of people. Namely, it's credited for Watson and Crick, but also Rosalind Franklin played a really big role. And it's a really sad story about how basically she didn't receive the proper credit because she died from doing the research that she provided to these guys, and they basically stole it from her. Pretty sad. But anyway, so DNA, really important, major discovery. It was the genetic code of life. It's like the blueprint that makes you what you are. And if you remember back to our session from last week, which none of you remember, oh, yeah, you were. Yeah. That's my only repeater. Anyway, if you remember back to there, we said the monomer that makes up DNA is called nucleotides. And nucleotides come in a few forms. They come in really 5 varieties, but for DNA, they come in 4. So there's a G, a C, an A, and a T. So G for guanine, C for cytosine, A for adenine, T for thymine. We're going to talk about these things called Chargaff rules, which not long after the discovery of DNA, we noticed, well, DNA has unique little features, little components to it, and they're found in certain ratios. There's a balance of how often we find these molecules. And generally speaking, these are the rules. G always pairs up to C, and A always pairs up to T. Okay? There's also different shapes to these molecules, which we call purines and pyrimidines. So I will once again reintroduce you to G-Cat.
Oh, yeah.
The most gangster kitty cat you've ever met in your life. And oh boy, here we go. We got to get some flavor going here. That's a kitty. That's a kitty. Don't judge me. And he wears this, he's a gangster, so he wears this big gold chain necklace with his own name on it, which you cannot get more gangster with. Anyway, GCAT is really, really helpful because he tells you a few things. The order of those letters really do matter. First off, G and C pair together, A and T pair together. But also, what sound do kitty cats make all the time? They purr, right? So since they purr, G, the first letter, means it's a purine. Oh, snap. And here's the other rule that Chargaff kind of discovered. Purines always pair up with pyrimidines. So that tells me if G and C pair together, then that means C is also a pyrimidine. Okay? And then we just repeat it. So A is also a purine, and T is also a pyrimidine. Hey! So now we can identify whatever is a purine or a pyrimidine because GCAT told you. And you always listen to what GCAT says. So if you know the rules for DNA, you're feeling pretty confident. Also, just to remind you, DNA has this like double helix structure, right? Oh, that's...
Masterpiece.
I'll sell that later on like eBay or something. But anyway, it's like this double helix structure, which basically means like a twisted up ladder, right? That twisted up ladder has these bonds that on the rungs of the ladder, like the steps of the ladder. There's 2 sides to it. If there's a G on one side, there's a C on the other. If there's an A on one side, there's a T on the other. And it looks like a random sequence of these things, and there's hundreds of millions of these in your DNA, but it's not random. Actually, the whole structure is super duper crazy important. But anyway, then there's the rules for RNA. Okay, RNA uses one different nucleotide. Yeah. It's G-cow. So once again, I know like female cows don't really have like horns like this, but I don't care. It's the most gangster cow you've ever met, everybody. It's G-cow. Remember, there's no he because it's a cow. It's a she. But anyway, GCAU. So it's the same rules. G and C pair together, and now A and U pair together. U now replaces the molecule thymine. So it's uracil instead of thymine. It structurally is the same thing. It's still going to be a pyrimidine. It's just the different quantity found in different things. RNA only has one side of that structure. It's also much shorter. DNA is really long, but RNA is much shorter. It's temporary. It's only used basically to do a few things, unless it's like a special kind of RNA. But, well, we'll talk about that maybe a little later. So we know these rules here. We're familiar with how they pair up. And then we go through a few processes of basically making these genetics work out. I'll walk you through like the basics of genetics first before we get too crazy into this, but at the end I'll tag it on there. But anyway, so we've mentioned all this to tell you that the structure of this DNA gets broken up into sections called chromosomes. Chromosomes basically look like this, and this is all one giant string of DNA, I should just mention to you. Okay. All right. Here we go. All right, it looks like that X shape, okay? And what this really is, it's two halves that are kind of split like this. Let's change colors, make life easier. These two halves, the halves are called chromatids. Yeah, so both halves are chromatids, so that's this whole thing here, okay? And then, of course, there's this middle region right here, which we call the centromere. It's merely the center. Yeah, and then there's these different length arms. This is called the P arm, which I believe stands for petite arm, or at least that's how I remember it. And then there's the Q arm, which means quite large. I don't know. I really don't know what it stands for, but it's big. It's the longer arm of the thing. When I was mentioning the kids getting ripped in half, those are the kids, right? Those are those X shapes, and each half chromatid is a kid, so it gets ripped in half. I know, right? And slightly offensive. So we've got the chromosomes. We know what those are. We've got DNA. We've got RNA. I just also want to throw this your way. We call this the central dogma. The central dogma. We got cats and cows and now dogs. They're everywhere, right? So central dogma. This basically means how information flows in genetics. First off, information always starts out in the form of DNA. And then we usually transition, we transition that to RNA. And then RNA gets transitioned, not much better, to protein. Honestly, the story of our cells is generally focused on trying to make proteins for your body because they're the most important molecule because they do everything. Literally, there's not something they can't do. If you're blinking, you're using proteins. If you're digesting food, you're using proteins. We talked about enzymes. We've talked about all kinds of messy things that happens to the body, and that's all thanks to this. So whenever we have DNA and we're trying to make more DNA, what do we call that? Replication. Good call. When we turn DNA into RNA, there's a word for that too. What do we call that? Transcription. It's a fun word. I don't know what it means, but that's a good one. But transcription. And then when we turn RNA to proteins, we call that translation. Okay? And we'll dive deeper into those later in the review. But for now, you should just be aware the central dogma is how we move things. So we've already talked about DNA replication, right? We mentioned that. We said that happens during synthesis stage in the cell cycle. But transcription and translation they're really, really important because this basically means your body can read your blueprint and then make a new version of that. It doesn't have to make an entire copy of your genetic information to make just one kind of cell or to make one protein that's found in your body. The analogy I used is like, it was around my birthday that we were talking about this, and I told you guys, like, my favorite kinds of cookies are Toll House cookies. Like,
What was your grandma's cookies?
they're great. They're wonderful. But they're variations of Toll House cookies. Anyway, so Toll House cookies, right? They're like beautiful, delicious chocolate chips, and they're like soft and thick and gooey. Oh, man, I'm just talking about food all morning now. I hope that makes you guys hungry. Chipotle, you're welcome.
I've only ever
Chick-fil-A,
gotten a small soda.
you're welcome. What did you say?
I've only ever gotten a small soda from Chipotle.
You know they have food?
Yeah, I know they have food, but I've
You should
only-
try it sometime.
I should.
It's pretty good. Anyway,
What is food?
it's that stuff you ingest to keep you alive, man.
You mean water?
Yep, that's what I meant. And I know you just stay alive by drinking water and staring at the sun.
That's because he's a swimmer.
Yeah. Yeah, you are. Anyway, so transcription translation basically gives our body a way of doing things. So I was saying, like, I love Toll House cookies. There's a recipe for the cookies. Now, my grandma makes a very special kind of cookie. It's got the right kinds of changes in the recipe that make it extra delicious. It's extra gooey. It's perfect. And she's written down that recipe in the family cookbook. Well, if I want somebody to make me cookies for my birthday, only grandma has the copy of that recipe. So you would need a copy if you're going to bake me the perfect cookies. So if you're going to go and get it, you're not going to be able to take the book home. That's my family's family cookbook. No, you can't take that. But you can maybe copy down the recipe real quick, and then you can take that recipe to your kitchen, get all the ingredients, put it together, bake them, and give them to me and make me a happy person. Well, that's the story of how this is made. That is the central dogma. The recipe is the DNA. The cookbook is the DNA. It's every recipe known to man, at least in Grandma's house. And then RNA is the short version, the RNA, the recipe version, and then the protein is the actual cookies. So that transition is what we see there. So transcription, translation. And once again, RNA is short. It's temporary. You could throw that recipe away and not hurt anybody's feelings. You can even bring it in the kitchen, get it all dirty and use it up and get cookie stains all over the thing. That's fine. So DNA to RNA, RNA to protein, and we know what those structures are. So knowing this, we talk about these things getting split up, sent around. So let's do this thing. Genetics. It was a big one. This phrase here describes when people inherit traits. So we use a few phrases, and we're going to use these same terms for all these examples. If somebody remembers, we've got these phrases dominant, and we've got recessive. Right? Dominant and recessive. Dominant is whenever an allele, it's a type of allele, which just basically means a variation of a trait. An allele that is expressed even if there's one copy. So we use letters to describe these alleles, and let's say in this case, dominant is going to be capital letter A. Recessive is an allele that is only expressed if two copies are together. Right? So in this case, we'd use the little letter a lowercase a. So when we see people being expressed, they have either a dominant appearance or a not dominant appearance. Now, an appearance, we use this phrase called phenotype, which means physical appearance or physical trait. Genotype describes, we'll just say the genetic alleles that the person has. And then we can phrase this in a few ways. So these letters combine. Alleles always come in sets of 2. So we can have big A, big A, big A, or little a, little a. There's no other ways to combine those letters. You might be thinking, well, what about little a, big A? What if the little letter came first? Well, we'd actually just move them and we'd swap them so that the big letter always comes first. So there's only 3 possibilities here. So when we're describing somebody's genotype, we can describe it in 3 ways. We can say this person here is homozygous dominant, which basically means homo means same, and we're basically saying these 2 same alleles are found there. And what shape are they? Are they dominant or recessive? We're saying they're dominant. In this case down here, we'd call this heterozygous. We don't have to say heterozygous dominant or recessive because we know it means one big, one small. And what's going to win? Dominant always wins. Because if dominant is paired with a recessive, the dominant always shines through to some degree, to some capacity. And then we have little a, which is going to be homozygous recessive. So what are they going to look like? What's their phenotype going to be? Well, this one is going to look dominant. This one is going to look dominant. And this one is the only time we'll see recessive actually show up in the population. So, basic terminology here out of the way, this goes back to this guy a long time ago, a monk named Gregor Mendel. Do you guys remember him? And he studied pea plants. Do you remember that? Now, pea plants are really unique because basically they can have a trait that is shown one way or they can show it the other way. So they only have a dominant or recessive version. Real genetics, like real detailed genetics, is way more complex than this. Like your hair color isn't the same between people. Like I have brown hair, but other people might claim they have brown hair and it's still a different shade than mine. And that's because the genetics of this have more genes turned on or activated to express that color in a different way. Skin colors, just like that. There are people that might identify as a certain race, but there's different tones to their skin color because skin color is very diverse in terms of the genetics that are there. So we are looking at the most basic form of genetics. It's very simple, but it's true in pea plants, which is just total circumstance that Gregor Mendel worked with pea plants that by chance had these traits. So let's go ahead and look at an example that he did. He looked at, and he didn't use Punnett squares like we are here, but he did this. He took a plant that he knew was purebred for the dominant trait. In this case, it's purple flowers and white flowers. So let's say purple is dominant and white is recessive. So he's got big A, big A. He knows they were purebred because he bred them himself. This purple flower had parents that were both purple, and their parents were purple, and their parents were purple. And he went back multiple generations to ensure the purple trait is only going to be found here. He did the same thing with a white flower and put it off to the side too. And then what he did is painstakingly, using a paintbrush and pollen from each flower, cross-pollinated them so that these purple plants mated with these white plants. And when he did that, he saw something interesting show up. Does anybody not know how to use a Punnett square? Well, just to remind you, just in case, all I did was I took each allele from each parent and I put them on different sides of a 2-box or 4-box square. And I'm just pulling from the top and moving that down, pulling from the top, moving that down, putting it in each box, and then pulling from the side, pulling from the side. So I get a big A and a little a. I put big A first because the capital letter always comes first. Anyway, so I do that, and every single box here makes a heterozygous individual. And what color would a heterozygous be if dominant is purple and white is recessive?
Purple.
It would be purple. Good job. Yas.
Purple.
Okay. So then he said, whoa, I just bred a purple and a white flower together. Where'd the white flowers go? What happened to that trait? So what he did is he crossed together the 2 of those kids. I know that sounds gross, but they're flowers, so it's cool. And he bred together these 2 heterozygous individuals. So when he did that, he crossed it up, and he crossed a big A with a little a, and a big A with a little a. And then in this first box, he gets big, big, big little, big little, little little. In this case, he saw the white trait show back up. What percentage of these kids are born white?
25.
It's 25%. 1 out of the 4 boxes, 1 out of 4 is 25% of all the boxes here. And 25% are little a, which have the white flower. He was able to make a discovery. Holy crap, traits can kind of hide. And that was his discovery, that there were dominant and recessive traits. People used to just think, oh, it's always a blend of the two that results. But he was like, nah, we're saying some of those traits show up more dominant than others. And that's what his big discovery was. So through that whole process, he also realized, or at least we realized today, that another 1/4 are going to be big A, and 2/4 or 50% are going to be big A, little a. But these together, I don't know what I just drew right there. Let's try this. These together, 75% are going to be purple. So it still shows, even though that dominant allele's there and the white still shows up, dominance still is taking over. And so that means some traits will become more common than others, and it can become predictable. So everything that we just described here, all those simple rules, we're going to kind of consistently use those, but there's a few tweaks and changes because we've learned over the years, well, genetics can be inherited in a few different ways. So, do you guys have any questions on complete dominance? Feeling okay? You sure? Okay. So the next version that we want to talk about is incomplete dominance. Incomplete dominance means it doesn't finish dominating. So if there's like, in the cases that we used in class, there's red flowers and white flowers, and if, let's say it's a snapdragon flower. Well, sometimes there are pink flowers. So what we're saying for incomplete dominance is that there's now a new, let me try that again. There's now a new third phenotype for heterozygotes. And this third phenotype, it looks blended. Okay, so it's a blend of the two different colors. So let's go ahead and say this. Let's use the capital letter R to describe red. Let's use the lowercase r to describe white flowers. And then we'll say, if we ever see a heterozygous, big R, little r, we'll see a new blended color, which would be pink. So it's kind of red, but the white's still kind of poking through. So it's kind of taken over. So that's what incomplete dominance is. So let's go ahead and say we do like what Mendel did. We take a red flower, and we cross it with a white flower. How do I figure out what the results are going to be? Yeah, I got to use a Punnett square. Ugh. Let me try this again. Oof, I'm bad at drawing. I'm very bad, and that's okay. You're very nice, but we all know I am.
Not that bad.
See? So, red and white. So I've got a parent. Remember, we always have 2 alleles for each parent, unless there's like a multiple allele trait. And then we've got 2 little r's for the white flowers. So big R's and all little r's. If I cross that up in this box, I'll get a big R and a little r, a big R, little r, a big R, little r, a big R, little r. This is going to basically say 100% of these kids are going to be what color? Pink. Why pink? Because we said they have a new third phenotype, and that phenotype is going to be unique. But that's going to say 100% of these kids are pink. If I were to cross this again with a pink and a pink, well, hopefully you could figure out what's going to happen. We're going to need to make a Punnett square. And by the way, they will give you scratch paper to be able to use in your EOC. I don't know how complex their questions can be, but if you can understand these different forms of inheritance, you should be able to answer almost anything that comes your way. So you got a big R, a little r, a big R, and a little r, and then in these boxes, we end up with 25% being red, 50% being pink, and 25% being white. You guys see how that is? Pretty common. I don't know how common it is in humans, but in terms of plants, it's pretty common. Yeah. So incomplete dominance, doing all right? Any questions there? No? So the other form of inheritance we talked about is called codominance. Codominance means it like gets along. So in this case, we mean two dominant alleles happen at the same time. That's pretty crazy. So in this case, some flowers do this too. We've got R, which is going to be red, W, which is going to be white, and R and W, which you notice both are capital letters. This is going to be both red and white, meaning there are like red and white splotches on the flower. So if you've ever seen like any flower with that kind of characteristic, that's a form of codominance. Anyway, so when we look, if I cross a red with a white, I get R and W for each one of these, which tells me that all of these kids 100% will be both red and white. I should also go ahead and take the moment to point out blood typing. Do you guys remember blood types? Blood type can get pretty messy, but it's a real world example. I'm trying to draw straight lines. That was me trying to draw straight lines. I'm gonna say it's fatigue. That's what it is. I'm just tired. That's it. Yeah. Yeah, it's so late today. It's like almost 11 a.m. But 11. Okay, so blood type. All right. Generally speaking, I use just all capital letters here, but we don't really use that format when it comes to like official ways. So I just want to talk about blood typing because they may reference that as an example. And in most cases, they use the capital letter I to talk about this protein that's like an antigen that describes blood type, right? So they use this capital letter I, and that's for dominant traits, and then there's a lowercase I. So if it's big I, little I, you guys know how to do that. You can do complete dominance, you get it. But I can come in a lot of variations. You could have an I with an A or an I with a B. So this leads us to a lot of different blood types. There's A-type blood, B-type blood, AB-type blood, and O-type blood. So I is going to, lowercase I is going to equal O-type blood. Basically meaning, oh, you don't have any of these components. You have nothing on you, which actually does mean you're extremely special. But anyway, let's say for A-type blood, we can have this in two versions. We can have IA with another IA. I know you might be getting confused because you're like, don't we only use two letters? Well, we only care about the I's to show us the letters, but then we're saying there's different types of I's, which is why these have little A's on there, or capital letter A's on there. Or you can be capital I with an A and a lowercase I. And then when we do B-type blood, it's the same thing, but we use only the letter B. And then there's AB-type blood, which means the only way you can have this is if you have both an A and a B. There's no other way to make this work. And if you're like, well, what about B and A, huh? Well, A comes before B in the alphabet, so if it comes B before A, we just swap them. And then O-type blood, the only way you can have that is if you don't have any version of this, which is little I and little I. All right? So let's just say, can we predict what a what a person would look like in terms of blood type by using these things? Well, yeah, absolutely. We'd still make a Punnett square, still the same kind of Punnett square. And let's say we take a person who's homozygous for A-type and they're heterozygous for B-type. Okay? Homo means same, so the two alleles have to be the exact same thing. And hetero means they're different. So that means they have one dominant and one recessive. So homozygous A means big I, and both of these must be like this because they're the same. Hetero B means they're different, so big I, little I, and then the B tells me the capital I has a B on it, which gives me I know I'm writing real tiny in there, and I apologize, but I'm trying. He's trying. So we've got AB type blood, and that's 50% of these kids. And then I've got A type blood, but they're heterozygous. That's also 50%. So this actually explains why some people that are born may not have the same blood type as mom and dad. In this case, there's a 50% chance that any kid born to a parent that's this way would have AB type blood, but mom has A type, dad has B type. It's not like some evidence that's like, hey, suddenly you find out somebody's not your mom or not your dad. You know what I mean? Because it is definitely a thing. So that's basically how blood type works. It's another form of codominance that incorporates some of the rules of complete dominance in there. How you guys doing? Yeah? Okay. Let's just get sex-linked traits out of the way. When we talked about chromosomes earlier, we said that you have 46 chromosomes or 23 pairs of chromosomes. And basically, they're really big and they kind of work their way down to being really small, right? And so we usually number them 1 through 23. And the smallest ones, actually, the smallest ones are really number 22. I take that all back. It's 22. But the last one, the 23 paired, is what we call the sex chromosome. And these can come in two forms. There's either an X or there's a Y. The X chromosome's like big, but the Y chromosome's really tiny. It's really small. It's so small, in fact, it can't contain a lot of genetic information. It can't contain the same amount, at least, as the X chromosome. I've heard it can only hold like 40 or 400 traits, which is really, really small in terms of chromosomes. So, females are genetically speaking XX, and males are genetically speaking XY. So females have two full sets of information. Males can only have one full set and one like partial set, okay? So knowing this, it means some traits can only fit to the X and won't fit to the Y. So that means we have to ignore the Y in some cases. Color blindness is a really good example of this. It's a recessive trait, so little b is going to say they are colorblind, okay? Capital B is going to, of course, tell us that they have normal vision. So if we cross a male with a female, which is, you know, sexually speaking, the only way that technically works, I mean, it's just genetics. I know we're not talking about gender roles. We're talking about just biological sex. So we've got XX and XY. And the X's here can have different traits. So let's say we've got a female, and let's say that female is colorblind. I know that's rare, and I'll explain why it is rare. And then we've got a male who is normal vision. He's a normie, okay? So if the female is colorblind, she has to be homozygous for colorblindness because it's a little b and a little b. It's the only way a person would actually express that trait. Whereas the male, he has normal vision. He has a capital B, but he has nothing on the Y. It's blank. We don't put any information there. So we got an XX, an XX, an XY, an XY. Notice how I did those capital letters first. Both of these sets of X's will have a capital B and a lowercase b, a capital B and a lowercase b. You have a colorblind mom, but in this case, there is a guarantee that all the females born to this mom will have normal vision because dad gave his X chromosome to make it a daughter, and his X chromosome has normal vision. So if it's a dominant trait, it takes over the recessives, and that's why it's harder to find females as colorblind. Down here on the bottom, an interesting thing happens, though. The X chromosome only has a little b, and this X chromosome only has a B. We put nothing on the Y, which means whatever that X chromosome has, the male has to express, because that's the only genetic information they have. So in this case, if a male is born to this couple, it's a guarantee that they will be colorblind. So this is an interesting scenario where, you know, you'd think, oh, I have a 50-50 chance, but now in this way, if you're born a female, you're good. You're just a carrier for colorblindness, but you don't have colorblindness. And male, in this case, definitely has colorblindness. There's no exception. How you guys doing? Any questions there? Fantastic. Okay, let me get the tricky ones out of the way, and then we'll like briefly mention pedigrees, and then you guys can go home. Or I guess you could go home anyway if you wanted to. But anyway, dihybrid is when we do something interesting. Instead of just having big A, this is when we add letters to it. So we're saying two traits at the same time, dihybrid, two hybrids, right? So two together. So what we can do to solve this is we could, and I'm rushing and it's getting really ugly. Big A, big B, little b. We could make a giganto Punnett square. And we'd just basically have to double everything. So we'd make a 16-box Punnett square, which would be crazy, but we could do it. If you wanted to take 10 minutes to do this thing, sure, go for it. Knock yourself out. But I'm going to be honest with you, don't do this. This takes too much time. If you're taking an EOC and they ask you a dihybrid cross question, which I doubt they will, because it just takes too much time, well, this is how you'd quickly figure things out. And also just kind of test that idea of complete dominance again real quick. Let's say I want to cross parents that's big A, and big A, little a. All I got to do is two separate Punnett squares. Much easier. Okay, and I just multiply my odds together. So if I'm crossing my A's, so this parent has big A, and this parent has big A, little a, I just do two little Punnett squares. Boom. Boom. I know that my odds for being big A are 1 and 2, and my odds of being big A, little a are 1 and 2. Then over here, I've got big B, I've got big B, little b, big, big, little, and then I look at my odds. Big B is going to be 1/2. Big B, little b is also 1/2. Cool, great, awesome, neat. So how do I find my odds if a kid is going to be big A, big B, little b? Well, I take my 1/2 for big A, and I times it by my odds of big B, little b, 1/2. So 1/2 times 1/2. 1 times 1 is 1. 2 times 2 is 4. So there's a 25% chance that the kid would look like that. Yay, we did it. Huzzah! You ready for pedigrees? Okay. Pedigrees kind of look like this. They have circles and they have squares. And they can be colored or half colored or not colored at all. So I wish I had like a paint tool that would just fill in things. I know there's probably a way to do it, but not on PowerPoint that I'm aware of. I'm trying to hurry. I'm sorry. So basically what this does is it looks for recessive traits. So the secret is this. Circles are female. Squares are male. And basically if they half color in, that means they're heterozygous. If they all the way color in, that means they're recessive for the trait. So same thing's true for males as well. And just to let you know, in this case, for these guys, we can call them hybrids, right? Just because it's a blend of the two. So if you've ever seen like a royal family tree, you know what I'm talking about? They're like, oh, your pedigree is very official. You're a royal, sir. And we actually do look at those family trees.
Is it supposed to be literally?
Oh, yeah, it is. I apologize. It's supposed to be little a. Good catch! Thanks for embarrassing me. It looks like a G. We're gonna blame somebody else. Okay, so we always start off with a male and a female. When we have a line that connects them like this, that means they had a baby, or maybe several. So the line that connects like this shows that they had sexual relations. Okay, and then we have a line that drops down, and let's say I'm trying to draw straight lines. I really am. Let's say this couple had 3 kids.
It looks like a spider.
It does. So let's say it's a female, the second one was a male, and the third one was another female. Let's say we're talking about color blindness. Why not? We know that's a recessive trait, right? And it's sex-linked, so that makes sense. Let's say that was a colorblind female, like we saw earlier, and we said that's a normal vision male. So he's all good, right? When we think about this, remember this is an X and a Y and an X and an X. That means this person has XX, this person has XY, this person has XX. By the way, we usually go, firstborn, secondborn, thirdborn. We go from left to right, but whatever. In this case, we can look at this and we can see, okay, this kid has to be at least a carrier. Remember? So we colored in halfway. For this kid, well, that X chromosome, they have to be at least a carrier because the X came from mom, and one X can only be a little b, and same thing's true here. So all of these kids are going to be carriers, so they'll look like this. If we want to, we can kind of show, like, there's this little relationship here. So let's say she met some dude, and he was colorblind. Uh-oh. Uh-oh. Right? And let's say they had 2 kids. Let's say 2 boys this time.
Oh, they all colorblind. Uh-oh.
Yeah. In this case, on the Y chromosome, well, actually, no. Remember, the Y chromosome doesn't carry anything in this case. But he's all the way colorblind, so we know his X is definitely colorblind. So there's a chance that the kid could be born colorblind, or there's a chance that he could be born normal. So we could say he's a carrier, or if we know for a fact he's colorblind, we'd color it in this way. The main thing is, can you see the family history now? By just looking at the shapes and how they're shaded in, you can see where recessive traits are. And we even did the more complex version by making it a sex-linked trait. But honestly, you could do it for just, recessive traits, which would be things like, a widow's peak or like earlobes attached to your face. You could actually trace the history that way. So with that, we're done.